How's everybody doing today? My name is Justin Esquivel. I am the CEO and Geneticist at Green Luster Phenos. I'll give you a little introduction of myself. My family and I are from Belize, Central America, just right outside of Corzal, but I've spent my entire life growing up around the cannabis plant. We started Green Luster Phenos in 2012 in Madrid, Spain, where we developed our first selective homozygous breeding facility out of Madrid, Spain. In Madrid, Spain, we practice axillary node culture, implementing apical meristem tissue culture. If we come across any pathogens or viruses like the hypoladen viroid or the mosaic virus, we can culture these viruses out utilizing apical meristem tissue culture. In 2016, we became federally licensed cannabis breeders in North Carolina, USA, where we also have a cannabis tissue culture laboratory, as well as a selective homozygous breeding facility. I have personally consulted on 76 cannabis facilities, 54 of which operate commercial canopies. We are now having our genetics grown in 76 different places in the world, 42 countries, and 19 different states. And so what we do in North Carolina is through apical meristem tissue culture, we are able to culture out the viroids within the genetic plants utilizing UVC exposure. All right, so without further ado, we're going to get right in this. We're going to talk about high revenue genetics. First bullet here, scarcity profile and marketplace value. So scarcity profile and marketplace value, this is so important because if you're a grower in the industry and you're cultivating a genetic that everyone is growing, you're going to get the lowest price point per pound for your genetics that you're growing. Second bullet, bred-in plasticities. This is something we're doing unique at Green Luster Phenos. We actually breed in resistances and plasticities to garden pressures, whether that's pest or bacteria. And we do this in-house by propagating fungus gnat larvae. We buy cells of powdery mildew and botrytis, and we practice what's called targeted releases. And so through our inbreeding program, we will select genotypes that show resistances to those specific plant pressures. Third bullet here, high cannabinoids and high terpenes. This is something you have to pay attention to when buying genetics. It's what allows the consumer to know what genetic they want to buy at the store. If you're dealing with high THC strains, your consumers are looking for 20% to 30% THC. And when it comes to total terpenes, they're looking for 3% or higher of total terpenes. Last bullet here, type 2 genetics. Type 2 genetics are genetics that are high in THC and high in CBD. Type 2 genetics have been our top seller for the last 2 years because a grower can grow type 2 genetics and get 2 turns of revenue on one harvest. These genetics will be, say, 16% CBD and 16% THC. So through chromatography and extracting, you can get 2 turns of revenue in 2 different products. These type 2 genetics are going to be compliant for CBD hemp here in the country of Czech, and they're also going to be the most vigorous, most robust, and the most resistant to your climate and regional zone. Cultivation success. Pictured here is a indoor grow with about 2,000 plants of our genetics. Clean cultivation practices is going to be the first topic I'm going to discuss. When growing in your environment, it's important that you wipe down anything that you're touching frequently with isopropyl alcohol to ensure that you have a sterile SOP. Weekly bacteria and pest control is imperative. You do this by putting out different products. I put 2 products up on the screen that I've used for the last 10 years with success. So you're going to implement integrative bacteria management, which is going to be the Xeritol HC product in blue on the screen. And then the second product in red, Lady Larve, is a highly concentrated water-soluble product that is put out and can be fed to your plants. It boosts your yields, your buds, and it also kills all the pests at their larvae stage. And canopy control utilizing a trellis. A lot of growers in the industry believe that a trellis is used to support buds, but it's not. A trellis is used to decrease plant height and overall increase light penetration. Defoliation of your canopy is also very important. Removing your big major fan leaves are going to allow you to have bigger yields and less of a chance to have powdery mildew or botrytis infect your canopy. Frost and flavor, this is probably my most favorite slide to talk about because I will get into some science of the plant. I'm going to discuss with you how you can naturally boost your cannabinoids and naturally boost your terpenes, doing so without spending any additional money or using any additional product. The first feminized seed ever created in the entire world was created by a process called rhodilization. Rhodilization is when a female cannabis plant is pushed past maturity and it is then reverted to a male, and it pollinates itself. This is a defense mechanism that's built into every female cannabis plant in the world. So all female plants, whether you get a clone or a feminized seed, they will all revert to a male. They'll all turn into a male if they're pushed past their maturity point. During this process of rhodilization, the female cannabis plant will produce double the cannabinoids and double the amount of terpenes. And so as a grower, the way you can implement this is dropping your temperatures by 10 degrees the last 2 weeks in flower. This will trick the plant to thinking it has reached the end of its flowering cycle, and it will increase trichomes and terpenes. And so having this in mind, everybody here needs to understand that the reason a female cannabis plant has pistils and the reason the female cannabis plant has trichomes is for the pollen. It's so when neighboring pollen blows by, it sticks to the plant and retracts down the pistil and forms a seed. Phytase is a new enzyme that we've been researching on a university level. We've done research and development with over 24 countries across the world with the enzyme phytase. And what we've discovered about phytase is it breaks down phosphorus and makes it bioavailable for the plant to uptake almost immediately. And through this research and development, we've discovered a boost in cannabinoids and a boost in yields as well. So as a grower, you have 3 ways you can start. You can start from seed, you can start from a clone, or you can start from a rooted tissue in vitro. I'm going to talk about the pros and cons to all 3. The advantage from starting from seed is you have a radicle, and the radicle is that first taproot that comes out of the seed. There are certain hormones produced by the radicle that allow you as the grower to achieve the full genetic potential of the genetic you're growing. The disadvantage of growing from seed is the instability. That's why clones exist. So obviously that is the advantage to growing from clone is you get the uniformity. However, the disadvantage is they don't have the radicle that has the hormones for you to achieve the full genetic potential. Talking about tissue culture, looking at this clone here, that's one clone, but utilizing tissue culture, you could get about 5 rooted tissues in vitro from that one clone. The other advantage of tissue culture that we like is that you don't ever have to water, you don't ever have to feed, and that plant will grow by itself in vitro for up to 1 year at a time. Here at Green Luster Phenos, my company, we implement axillary node culture and we implement apical meristem tissue culture. If we come across any pathogens or viruses, we implement a patented technology of UVC exposure. Tissue culture is a way that you can have up to 50,000 rooted tissues in vitro in a space the size of your bedroom. Your nodes go into small culture tube vessels like this. In these vessels, they'll stay 100% sterile, and they'll grow for up to 1 year. There's 2 different types of media. There's rooting media and there's multiplication media. Rooting media obviously produces roots. The multiplication media produces the vegetative growth. This process of tissue culture, I'm teaching commercial growers how to do it all over the world. We're also teaching home growers how to do it as well. It's very easy to learn. I have personally set up 27 tissue culture laboratories in the USA, and we've done 2 in Europe and 1 in Canada as well. And all of our commercial operators are telling us it's saving them about 70% of their monthly revenue. The reason why tissue culture is so important is because half of the growers I get in touch with, they think their plants have a deficiency, but there is no deficiency. They simply have the hop-laden viroid, which looks just like a deficiency. So if you're breeding plants with the mosaic virus or the hop-laden viroid, 9 times out of 10 you don't know. And we have personally tested tens of thousands of seeds and clones being passed all over the world that are testing positive for the hop-laden viroid. The biggest advantage to a grower on a commercial scale or a home grower with tissue culture is your favorite plant in your garden or your favorite genetic that you really, really don't want to lose. You can initiate into media and just leave it alone, and you can come back a year later, pull it out, and start growing it. So everything in the media is everything that the plant needs to survive. The food, the hormones, the nutrients, the sugars and sucrose that it needs, everything that plant needs to survive for 1 year is inside of those vessels. One thing that I did forget to mention that I want to say is with the type 2 genetics, a lot of people don't understand how CBD counteracts the psychotropic effect of THC. It counteracts all the negative effects, the dry eyes, the dry mouth, the blurred clarity. So smoking a genetic with high THC and CBD, you are going to have the best consumer experience with clarity. But thank you, everyone, and again, thank you to KONOPEX for having me out, and it's been great being able to visit out here in Czech.